Hello everyone, hope you're all doing well. I'm back with another topic that is Fimbriae and pili. So this is in the continuation of bacterial cell structure. Let's start with the topic. Fimbriae and pili are hair-like appendages present on the bacterial cell wall that are quite similar to the flagella, but they are shorter and more in number as compared to the flagella. I have recorded a lecture on the flagella before this, so if you haven't listened to that, you can go and check it out. Talking about the fimbriae and pili, they are involved in different functions like attachments and motility and also the bacterial conjugation. We can use these terms alternatively, but if we talk about the bacterial conjugation, we can say that pili or the sex pili perform this function. Bacterial conjugation is done through the help of pili. So both the pili and fimbriae are present in both gram-positive as well as gram-negative bacteria, but they are more prevalent and commonly found in gram-negative bacteria. Talking about the structure and composition, they have thin protein tube structure that is originating from the cytoplasmic membrane of the bacteria, which help the bacteria to stick to the surfaces. But if we see the difference that pili are typically longer and fewer in number as compared to the fimbriae. And both the fimbriae and pili have the shaft composed of a protein called pilin, and in case of fimbriae you can call it as fimbrian as well. And the end of that shaft, there is an adhesive structure or the adhesive tip that have a particular shape which corresponds to the specific glycoprotein or glycolipid receptor on the host cell. It will get clearer to you with the help of this image that there's a shaft protein which have this adhesive tip, and this tip corresponds to this structure which having the glycoprotein or glycolipid which is present on the host cell. So this host cell contains this tip receptor, and this adhesive tip of the pillars or fimbriae gets attached to it as they have the corresponding shape as well. Then fimbriae are approximately thousands of fimbriae are present on the bacterial cell, but we cannot see them with our naked eyes, and they are only visible through electron microscope as they are very tiny minute structure, and they are in the form of cylinder tubes that are helically arranged protein subunits, composed of helically arranged protein subunits, and they are about 3 to 10 nanometer in diameter with several micrometers long. So fimbriae are the long structures of several micrometers having the diameter of 3 to 10 nanometer. So some types of the fimbriae help the bacteria to attach to the solid surfaces like rocks in streams and host tissues. For example, if we talk about the stream or the moving water and the bacteria is supposed to attach to a surface like rock, the fimbriae will be the structure that will help in this attachment of the bacteria to the rock surfaces. Alright. Then type 4 fimbriae are present, they could be present at one or even at the both poles of the bacterial cell, and they also help in the attachment of the bacteria to different objects, and this is required for the twitching motility that occurs in some bacteria. So these type 4 fimbriae are found in some bacteria like Pseudomonas aeruginosa, Neisseria gonorrhea, and some strains of E. coli also have this. Then pili, they are lesser in number as we have talked before. The sex pili are approximately 1 to 10 sex pili are present per cell, bacterial cell. Their singular is pilus, or the plural is pili. So 1 to 10 sex pili could be present per bacterial cell. And there are also the hair-like structures that are quite different from fimbriae in the following ways. That pili are often larger as they are 10 to 9 to 10 nanometer in diameter, as we have talked about the fimbriae, which is 3 to 10 nanometer in diameter. So they are often larger. Pili are often larger than fimbriae. Then they are genetically determined by conjugative plasmids and are required for conjugation. So one of the basic, or you can say a unique function that the pili does as compared to the fimbriae is this conjugation. So these six pili are involved in the conjugation, which help in the transfer of the extra chromosomal factor or the plasmid from the donor cells to the uh, recipient cells. Then some bacterial viruses attach specifically to the receptors on sex pili at the start of the reproductive cycle. So in the start of the reproductive cycle, some bacterial cells have um, this ability to attach to these specific receptors that are present only on the sex pili. Uh, so this also happens, or we can say one of the function or property of pili. We have two types of pili. You can say one is a long conjugative pili and the other one is short attachment pili. As you can tell from the name that the long and the conjugation pili helps in the process of conjugation, and this short pili will help in the attachment. Short attachment pili, which are also known as fimbriae. So as I told you before that we can use these terms alternatively as long as we're talking about the attachment of the bacteria. So these are usually short and quite large in number. So they're numerous in number but usually short. They enable the bacteria to colonize on some surfaces, environmental surfaces, or cells, and they also resist flushing, means they are not be easy to be removed of. Alright, so the function of these short pili or the fimbriae is the attachment of the bacteria cell to the surfaces and resistance of these bacteria from flushing. And the long conjugation pili, which are also known as F or sex pili, they are longer but fewer in number. So the short attachment pili or the fimbriae are shorter but larger in number, but the conjugation pili are longer but shorter in number. Alright, so what happens in this conjugation? So conjugation pillars enables conjugation. So conjugation is the transfer of DNA from one bacterium to the other by cell to cell contact. So two cells come in contact with each other with the help of this pillars, and they will help in the transfer of the bacterial DNA or the F plasmid, that is the fertility plasmid, from one donor bacterial cell to the other recipient cell. So in gram-negative bacteria, it is typically the transfer of DNA from the donor or the male bacterium with the sex pilus. So the male bacterium will have the sex pilus that will get attached to the recipient or the female bacterium, and their genetic material or their DNA or maybe their F plasmid will get transferred from one bacteria to another, and this could happen through genetic recombination. Alright, so here you can see there is a donor cell with the pilus, and this is the shutter plasmid, or you can call it an F plasmid, and this is the recipient cell with no plasmid, or you can say it is F negative, or if it doesn't have any fertility plasmid. So what will happen, this pilus will form a pathway through which these two cells come in contact with each other, and their plasmids will get transferred. So now both these cells are F positive now, and both these cells have transferred their genetic material, or though it was an extracellular chromosome, it was a plasmid, or you can call it has a fertility or F plasmid that has been transferred from the donor cell to the recipient cell. So now looking into the difference between the fimbriae and pili according to these parameters. First of all, the structure, which is one of the parameter, in which we can see that the fimbriae are more blister-like fibers, and the protein is commonly known as fibrin, whereas the pili are thin or fine hair-like microfibers with the protein pilin forming a thick tubular structure. So the fimbriae are more blister-like fibers, and the pili have more hair-like microfibers. So you can see the difference is more in ultrastructure. Then in length, the fimbriae are shorter, whereas the pili are longer. And talking about the occurrence, occurrence of the fimbriae is more per cell. So each cell could have 200 to 400, maybe up to thousands of the fimbriae, whereas a one cell can contain 1 to 10 sex pili. Alright. Then both the gram-negative and the gram-positive bacteria have fimbriae, whereas the pili are more commonly found or are more prevalent in gram-negative bacteria. Talking about the rigidity, the fimbriae is a less rigid structure, whereas the pili is a more rigid structure. The formation, how they are formed. So the formation of fimbriae is due to or with the help of the genetic material of the bacteria. So it is administered by the bacterial genes in the nucleoid area. So the formation is with the help of the bacterial genes, whereas the pili, its formation is administered by the plasmid genes. So the genes for the formation of pili will be present in the plasmid region, whereas the genes for the formation of the fimbriae will be present in the bacterial DNA. And one of the major difference in this function is that fimbriae plays a role in attaching cells to the surface. Although pili also do that, but more commonly the sex pili, or the difference we can say that the difference that is created in the function is that the pili are involved in the bacterial conjugation. All right, then fimbriae versus cilia. So we can say that all these hair-like structures, but why we give them different names? But we can see like we have a difference between the fimbriae and the pili and the flagella. So similar is the case with the cilia. They're also the hair-like structure, but what makes them different is that they are present in eukaryotic cells. Alright, so fimbriae are present in prokaryotic cells, whereas the cilia is present in eukaryotic cell, where they are responsible for the locomotion or the movement of the cell. But if we talk about the fimbriae, their function is not only about the motility, but they are more importantly are involved in the attachment of the bacteria to surface. So the attachment of the bacteria to the surface is done by the fimbriae, but the major function of the cilia is the locomotion or the motility of the cells, and also that they are present in the eukaryotic cell. So this is it for today's video. Thank you so much. Hope you like it. Please like and subscribe this channel if you want to see more such videos. Till then, goodbye.